Tonight, we're going on a journey through something you use every single moment of every single day, yet rarely stop to consider. Your senses, those remarkable systems that transform the physical world into the lived experience of being human. We're not just talking about the famous five, we're diving deeper into hidden senses you didn't know you had, into the strange and beautiful ways your brain weaves together touch and taste and sound into a seamless experience of reality. You'll hear about people who can taste colors, about the receptor cells that let you know exactly where your hand is even in total darkness, and about the way a single scent can pull you back through decades to a memory you thought you'd forgotten. So settle in, let your breathing slow, let your body relax into wherever you're resting tonight, because for the next little while, we're going to explore the quiet miracle of perception itself. And if these calm explorations help you unwind, if they give you something gentle to think about as you drift towards sleep, consider liking and subscribing to Whispered Knowledge. It helps others find their way here too, one curious mind at a time. Now, let's begin. Vision, the dominant sense. Let's start with the sense that dominates so much of human experience. Vision. Roughly 80% of the information your brain processes comes through your eyes. That's an extraordinary proportion, and it speaks to how central sight is to the human experience of the world. Your eyes are extraordinary instruments, each one containing over 2 million working parts. That makes them second only to the brain in complexity among your organs. And those parts work together with astonishing precision every moment you're awake, processing roughly 10 million bits of information per second. Light enters through your cornea, that clear curved window at the front of your eye. The cornea does most of the focusing work, bending light rays by roughly 2/3 of the total focusing power needed. It's also one of the few parts of your body that has no blood supply. It gets oxygen directly from the air and nutrients from the aqueous humor, the clear fluid that fills the front chamber of your eye. Then the light travels through your pupil, that black opening that expands and contracts depending on how much light is available. In bright conditions, your pupil can shrink to just over 2 millimeters in diameter. In darkness, it can expand to nearly 8 millimeters, letting in up to 16 times more light. This happens automatically, constantly adjusting without any conscious thought from you. The adjustment takes mere fractions of a second, orchestrated by the iris, that colored part of your eye that acts like a biological camera aperture. Behind the pupil sits the lens, a remarkable flexible structure that changes shape to fine-tune focus. When you look at something close, tiny ciliary muscles pull the lens into a rounder shape, increasing its focusing power. When you look at something distant, they relax and let it flatten. This process, called accommodation, happens so smoothly you never notice it. But it's happening every time you shift your gaze from this screen to something across the room, thousands of times per day. The lens is made of crystalline proteins arranged in such precise layers that it's completely transparent, even though it's living tissue. As you age, these proteins can become less flexible and less transparent, leading to presbyopia and cataracts. But in youth, the lens is so flexible that children can focus on objects just a few inches from their eyes, something most adults can no longer do. At the back of your eye lies the retina, a thin layer of tissue barely thicker than a sheet of plastic wrap, yet packed with light-sensitive cells and the neural circuitry needed to begin processing visual information. This is where the magic really happens. Your retina contains approximately 100 to 120 million rod cells and around 4.5 to 6 million cone cells. Though these numbers vary somewhat between individuals, arrayed in layers with astonishing precision. The rods handle vision in dim light, detecting movement and shapes when colors fade away. They're incredibly sensitive. A single rod cell can respond to a single photon of light, though your brain requires several photons hitting multiple rods before you consciously perceive light. Rods are distributed throughout the retina, but are most concentrated outside the central area, which is why objects are often easier to see in dim light when you look slightly to the side of them rather than directly at them. The cones, concentrated in a small central area called the fovea, give you sharp, detailed vision and let you see color. The fovea is only about 1.5 millimeters across, but it contains roughly 200,000 cones per square millimeter, packed so densely that they're at the very limit of what's physically possible. When you look directly at something, you're aligning it with your fovea, using this tiny patch of ultra-high resolution color vision. Those 6 million cones come in 3 types, each sensitive to different wavelengths of light. One type responds most strongly to long wavelengths, peaking around 560 nanometers, which we perceive as red. Another responds to medium wavelengths, peaking around 530 nanometers, which appear green. The third responds to short wavelengths, peaking around 420 nanometers, which we see as blue. Every color you've ever seen is created by the varying responses of these 3 cone types, combined and interpreted by your brain. It's why we call human vision trichromatic, 3-color. But here's something that might surprise you. You're actually colorblind in dim light. Those cone cells that detect color need relatively bright light to function. Below a certain threshold of illumination, they simply don't respond anymore. In darkness, only your rod cells are working, and they can't distinguish colors at all. That's why everything looks gray and washed out when you walk outside on a moonless night. The world hasn't lost its color. Your eyes have just switched to a different mode of seeing, optimizing for sensitivity rather than color and detail. Your eyes move constantly, even when you think you're holding them still. These tiny movements, called microsaccades, happen several times per second. They're essential for vision because your photoreceptor cells actually stop responding if an image stays perfectly still on your retina. This phenomenon is called troxler fading. By constantly shifting the image slightly, these microsaccades keep your vision fresh and clear. Without them, the world would literally fade from view within seconds. When you do move your eyes deliberately, scanning a scene or reading a line of text, your eyes don't glide smoothly. They jump in quick movements, called saccades, several times per second. Each saccade lasts only 20 to 200 milliseconds, and during that time, your eyes can move at speeds up to 900 degrees per second. During each saccade, your vision is essentially turned off. Your brain suppresses the blurry motion so you don't experience it, a phenomenon called saccadic suppression. That's why you can't see your own eyes move when you look in a mirror. Each time you try to catch the movement, your brain is already editing it out. Between saccades, your eyes pause for brief moments called fixations, typically lasting 200 to 300 milliseconds. This is when you actually take in visual information. Reading, for instance, is a series of fixations, with saccades jumping from one word or cluster of words to the next. Skilled readers make fewer, more efficient saccades, while beginning readers make more frequent, less targeted eye movements. The human eye can distinguish roughly 10 million different colors. That's an enormous range, created entirely by the varying responses of just 3 types of cone cells. Some people, though, may have even more potential. Studies suggest that perhaps as many as 12% of women carry a genetic variation that gives them a 4th type of cone cell, responsive to wavelengths between the standard red and green cones. However, having 4 cone types doesn't automatically translate to enhanced color vision. The brain must also be able to process the additional information, and this functional tetrachromacy appears to be quite rare. Scientists have identified only a handful of confirmed cases where individuals can actually perceive and distinguish colors differently because of their fourth cone. When functioning properly, this could theoretically allow perception of up to 100 million different shades, though exactly how tetrachromats experience the world remains an active area of research. On the other end of the spectrum, about 8% of men and 0.5% of women have some form of color vision deficiency. The most common type is red-green color blindness, where one type of cone is missing or doesn't work properly. People with this condition see the world in a different palette, with reds and greens appearing more similar than they do to those with typical color vision. It's not that they see in black and white. They simply have a different, slightly narrower color experience. Interestingly, some research suggests that people with red-green color blindness may be better at detecting certain types of camouflage, as they're less distracted by color differences and more attuned to brightness and texture patterns. Your peripheral vision is far more sensitive to movement than your central vision. That's because the outer edges of your retina have a higher concentration of rod cells, which excel at detecting motion and changes in the visual field. This is an evolutionary gift. Our ancestors needed to spot predators approaching from the side, even while focused on a task directly ahead. Today, it's why you notice a car approaching from the side street before you consciously turn to look at it, or why you see a bird flitting past in your peripheral vision even when you're reading. But peripheral vision is terrible at seeing detail or color. Hold your arm straight out to the side while looking straight ahead. You can sense that your hand is there, and you notice if you wiggled your fingers, but you can't count how many fingers you're holding up without turning to look. The further from the center of your vision, the more the detail fades away. This is because cone density drops off dramatically outside the fovea, and your brain doesn't devote as much processing power to peripheral information. Here's something that sounds impossible until you think about it. You have a blind spot in each eye, a small area where the optic nerve exits the retina, and there are no photoreceptor cells at all. It's roughly 15 degrees to the right of center in your right eye, and 15 degrees to the left of center in your left eye. The blind spot is about 7.5 degrees wide and 5.5 degrees tall, which at arm's length could hide something the size of a grapefruit, and yet you never notice it. Your brain fills in the gap so seamlessly that the blind spot is completely invisible to you. It doesn't just leave a hole in your vision, it actively fills it in with what it expects should be there based on the surrounding visual information. It takes careful testing with specific visual targets to even prove it exists. The human eye can detect a candle flame from nearly 2 miles away on a dark, clear night. That's how sensitive your vision is to light. And at the other extreme, your eyes can adapt to brightness levels that vary by a factor of 1 billion. From starlight to sunlight, your visual system constantly adjusts, managing an enormous range of light intensity through a combination of pupil adjustment, switching between rod and cone vision, and neural adaptation in the retina and brain. Dark adaptation, the process of your eyes becoming more sensitive in darkness, takes time. After about 5 to 10 minutes in darkness, your cone system reaches its maximum sensitivity. But rod adaptation continues for up to 30 minutes or more, gradually becoming more and more sensitive. This is why it takes so long for your eyes to fully adjust to darkness after you turn off the lights, and why even brief exposure to bright light can reset the adaptation process. Your eyes also process information astonishingly fast. Studies suggest the human brain can process images seen for as little as 13 milliseconds. That's faster than a single frame in most movies, which play at 24 frames per second. Fighter pilots have been shown to identify specific aircraft from images flashed for just a few milliseconds. Your visual system can extract meaning from the briefest glimpse, recognizing faces, objects, and scenes before you're even consciously aware of seeing them. Tears aren't just for crying. Every time you blink, you spread a thin layer of tears across your cornea. This tear film has 3 distinct layers, each serving essential functions. An outer oily layer, produced by the meibomian glands in your eyelids, keeps the tears from evaporating too quickly. A middle watery layer, produced by the lacrimal glands, provides moisture and oxygen to the cornea and washes away debris and microorganisms. An inner mucus layer, produced by goblet cells in the conjunctiva, helps the tears stick to the eye's surface. Without this constant lubrication, your corneas would dry out and vision would blur. You blink roughly 15 to 20 times per minute while awake, spreading fresh tears each time, which adds up to about 28,000 blinks per day. Each blink lasts roughly 1/10 to 4/10 of a second. That means you spend about an hour and a half per day with your eyes closed just from blinking. Yet you never notice the darkness during blinks. Your brain suppresses the visual input during the blink and also slightly adjusts your perception of time to edit out the gap, creating the illusion of continuous vision. And vision isn't processed by your eyes alone. The real work happens in your brain. The raw visual data from your retinas travels along the optic nerves, each containing over 1 million nerve fibers, to the visual cortex at the back of your brain. But before reaching the visual cortex, the signals pass through the optic chiasm, where fibers from the inner halves of each retina cross to the opposite side of the brain. This crossing ensures that each half of your brain receives information from both eyes about the opposite side of the visual field. In the visual cortex, the information is processed in multiple areas, each handling different aspects of what you see. Some areas analyze color, others track movement, still others identify edges, shapes, faces, or text. The visual cortex contains specialized regions like the fusiform face area, which is particularly responsive to faces, and area V5, which is specialized for detecting motion. Your brain assembles all these separate streams of information into the unified visual experience you call seeing, binding together color, motion, depth, and form into coherent objects and scenes. This processing is remarkably efficient, but not infallible. Optical illusions reveal just how much your brain interprets and constructs what you see. When you look at an impossible staircase or a dress that appears different colors to different people, you're seeing the consequences of your brain's visual processing shortcuts. Your brain makes assumptions based on experience, lighting, context, and depth cues. Usually these assumptions work perfectly, allowing you to navigate a complex 3-dimensional world from 2-dimensional retinal images. But illusions expose the seams in the system, showing you that what you see isn't quite the same as what's actually there. Your brain even invents visual information when needed. When you look at a neon sign where curved lines appear to glow between dots, you're seeing illusory contours. The glowing curves don't exist in the image. Your brain creates them based on the arrangement of the dots. Similarly, when you perceive depth from a 2-dimensional image or motion from a series of still frames in a movie, your brain is actively constructing perceptions that go beyond the raw sensory input. The visual system can also adapt to dramatic changes. People who wear inverting goggles that flip their vision upside down initially see the world inverted, but after several days of continuous wear, their brains adapt, flipping the image back so they see normally again. When the goggles are removed, the world appears upside down briefly, until the brain readapts. This demonstrates the remarkable plasticity of visual processing and the extent to which your brain actively constructs your visual experience rather than simply displaying raw input. Hearing, the world of sound. Now, let's shift to another sense that shapes your experience constantly. Hearing. Sound is invisible, yet it fills the space around you, carrying information, emotion, music, and meaning through the air. Unlike vision, which can be blocked by simply closing your eyes, hearing is always on. Even while you sleep, your auditory system continues processing sounds, alert for anything that might signal danger or require attention. Sound itself is simply vibration traveling through a medium. When something vibrates, whether it's a guitar string, a vocal cord, or a door slamming shut, it pushes against the air molecules around it, creating tiny pressure waves that spread outward in all directions like ripples on a pond. When those waves reach your ear, they set off a remarkable chain of events that ends with your brain perceiving sound. Your outer ear, that visible curved structure on the side of your head called the pinna or auricle, is designed to catch and funnel sound waves inward. Its complex folds and ridges aren't just decorative or accidental. They help you localize sounds, especially those coming from above or below. The shape of your outer ear creates subtle differences in how sounds from different directions reach your eardrum, adding small delays and filtering certain frequencies based on the angle of arrival. Your brain has learned to interpret these acoustic signatures, allowing you to determine not just whether a sound is coming from your left or right, but also from above or below, in front or behind. Sound waves travel down your ear canal, a tube roughly an inch long that ends at your eardrum, also called the tympanic membrane. This membrane is thin, barely thicker than a piece of tissue paper, and slightly cone-shaped. It vibrates in response to the incoming pressure waves. Every sound you hear begins as a vibration of your eardrum. A whisper creates tiny vibrations. A shout creates larger ones. But even the loudest sound only moves your eardrum about the width of a single hydrogen atom. The sensitivity required to detect such minute movements is extraordinary. The ear canal itself isn't just a passive tube. Its length and shape create a resonance that amplifies sounds in the 2000 to 4000 Hz range by as much as 10 to 20 decibels. This happens to be the frequency range that's most important for understanding human speech. Evolution has fine-tuned the mechanical properties of your ear canal to enhance the very sounds that matter most for communication. Behind the eardrum lies the middle ear, an air-filled chamber containing 3 of the smallest bones in your body. They have evocative names: the malleus, incus, and stapes. Commonly called the hammer, anvil, and stirrup because of their shapes. These bones, tiny enough that all 3 could fit on a penny, are connected in a chain that stretches across the middle ear cavity. The malleus is attached to the inner surface of the eardrum and moves with it. The incus connects the malleus to the stapes, and the stapes, the smallest bone in your entire body, measuring just 3 millimeters in length, presses against a membrane covering the entrance to the inner ear. This arrangement forms a lever system that amplifies the vibrations from your eardrum by roughly 22 times in force, though at the cost of reducing the range of motion proportionally. Why is this amplification necessary? Because the next step involves transmitting vibrations from air into fluid, and without amplification, most of the sound energy would be lost at the interface. Sound waves travel easily through air, but are mostly reflected when they hit a denser medium, like water or the fluid-filled inner ear. The middle ear's lever system overcomes this impedance mismatch, ensuring that most of the sound energy makes it into the inner ear. The middle ear also contains the smallest muscles in your body, the stapedius and tensor tympani. These muscles can contract to stiffen the ossicular chain, reducing the transmission of very loud sounds and protecting the delicate structures of the inner ear from damage. This acoustic reflex happens automatically in response to loud sounds, though it takes roughly 40 to 100 milliseconds to kick in, which means it can't protect against sudden, extremely loud noises like gunshots or explosions. The stirrup bone presses against a membrane called the oval window, which covers the entrance to the inner ear, where things get truly remarkable. Inside the inner ear is a snail-shaped structure called the cochlea, from the Greek word for snail. It's only about 10 millimeters in diameter, coiled 2.5 times like a spiral staircase, and filled with fluid. If you could unroll the cochlea, you'd find it's divided lengthwise into 3 fluid-filled chambers separated by membranes. The key structure is the basilar membrane, which runs the length of the cochlea and is lined with the organ of Corti, which contains around 15,000 to 16,000 hair cells arranged in precise rows. These cells are the actual sensors that detect sound. Each hair cell has dozens of stereocilia projecting from its top, arranged in order of increasing height like organ pipes. When vibrations reach the cochlea, they create waves in the fluid inside, and these waves cause the basilar membrane to move up and down. This movement bends the stereocilia on the hair cells. When the stereocilia bend toward the tallest one, ion channels at their tips open, allowing charged particles to rush in and triggering the cell to send an electrical signal to the auditory nerve. The basilar membrane is not uniform along its length. It's narrower and stiffer at the base, near the oval window, and wider and more flexible at the apex, deep inside the cochlea. This gradient of mechanical properties means that different frequencies of sound create waves that peak at different locations along the cochlea's spiral. High-pitched sounds with short wavelengths create waves that peak near the base, where the membrane is stiffest. Low-pitched sounds with longer wavelengths travel further along the spiral before peaking near the apex, where the membrane is most flexible. This is how your ear separates sound into different pitches through a mechanical filtering process that occurs before any neural processing begins. Each ear contains around 15,000 to 16,000 hair cells in total, divided into inner hair cells and outer hair cells. The inner hair cells, about 3,000 to 3,500 of them arranged in a single row, are the primary sensory cells. They detect the motion of the basilar membrane and send signals to the brain. The outer hair cells, about 12,000 of them arranged in 3 rows, have a different job. They can actually change length in response to electrical signals, essentially acting as tiny motors that amplify the motion of the basilar membrane. This cochlear amplifier can boost the response to quiet sounds by as much as 40 to 50 decibels, dramatically increasing the sensitivity and frequency, selectivity of hearing. The hair cells are extraordinarily sensitive. The quietest sound you can hear causes hair cells to move by less than the width of a single atom, roughly 1/10 of a nanometer. That's detecting motion at nearly the molecular scale. The fact that you can hear such tiny vibrations while filtering out the much larger random thermal vibrations of molecules in the fluid around the hair cells is a testament to the exquisite engineering of the cochlea. The human ear can detect frequencies from about 20 Hz to 20,000 Hz in young, healthy ears. 20 Hz is an extremely low rumble, felt almost as much as heard. 20,000 Hz is a high-pitched whine at the very edge of perception. Most of the sounds you encounter fall somewhere in the middle. Human speech, for instance, typically ranges from about 100 Hz to 8,000 Hz, with most of the acoustic energy and information content concentrated between 200 and 4,000 Hz. But you're most sensitive to sounds between 2,000 and 5,000 Hz. In this range, your hearing is so acute that you can detect sounds at threshold levels, as quiet as 0 decibel sound pressure level, or 20 micropascals. This happens to be right in the middle of the frequency range where human speech carries most of its information, especially the consonant sounds that are crucial for understanding words. It's no coincidence that your hearing is most acute right there. Evolution has tuned your ears to be especially good at detecting the sounds that matter most for communication. Your two ears work together to help you locate sounds in space through a process called binaural hearing. When a sound comes from your right side, it reaches your right ear slightly before your left ear. The difference might be only a few hundred microseconds, a delay smaller than the blink of an eye, but your brain detects it. Your brain also compares the loudness at each ear, since the sound has to travel further to reach the ear on the opposite side, and because your head casts an acoustic shadow, especially for higher frequency sounds. By analyzing these tiny differences in timing, called interaural time differences, and loudness, called interaural level differences, your brain can pinpoint where a sound is coming from with remarkable accuracy. In ideal conditions, you can localize a sound to within 3 to 5 degrees of arc in the horizontal plane, roughly the width of your fist held at arm's length. But this system has limitations. It's hard to distinguish whether a sound is coming from in front of you or behind you, since it reaches both ears at the same time and with equal loudness from either direction. Similarly, sounds directly above or below can be ambiguous. This region of confusion is sometimes called the cone of confusion. That's where the shape of your outer ears helps. Sounds from different directions bounce off the folds of your pinna in slightly different ways, creating subtle filtering effects that change the frequency content of the sound. Your brain has learned to interpret these spectral cues, allowing you to resolve front-back and up-down ambiguities. Interestingly, your outer ears continue to grow throughout your life, which means the acoustic filtering they provide changes slowly over time. Your brain adapts to these changes, but if you were to suddenly have someone else's ears attached to your head, sounds would seem to come from the wrong directions until your brain learned the new filtering patterns. Your ears aren't just for hearing, they're also essential for balance and spatial orientation. Inside your inner ear, right next to the cochlea, are 3 semicircular canals. These fluid-filled loops are oriented in 3 different planes, roughly corresponding to the 3 dimensions of space, pitch, roll, and yaw. When you move your head, the fluid in these canals sloshes around due to inertia, bending hair cells similar to those in the cochlea. But instead of detecting sound, these hair cells detect rotation and angular acceleration. Also, in the inner ear are 2 otolith organs, the utricle and saccule, which detect linear acceleration and the direction of gravity. They contain tiny calcium carbonate crystals called otoliths, ear stones, resting on a gelatinous layer over hair cells. When you tilt your head or accelerate, gravity or inertia pulls on these crystals, bending the hair cells beneath them. Your brain uses signals from the semicircular canals and otolith organs, along with input from your eyes and your sense of touch and body position, to maintain your balance and sense of orientation. When something disrupts this system, like an infection in the inner ear, the accumulation of calcium debris in the semicircular canals, a condition called benign paroxysmal positional vertigo, or conflicting signals from different senses, Like when you're reading in a moving car, you can experience vertigo, a spinning sensation that can be profoundly disorienting and nauseating. Tinnitus, the perception of ringing or buzzing in the ears when no external sound is present, affects roughly 15 to 20% of people to some degree. For most, it's a mild annoyance. For some, it's a constant, intrusive presence that can interfere with sleep, concentration, and quality of life. The causes vary. Sometimes it's damage to the hair cells in the cochlea, which can occur from noise exposure, Weijing, certain medications, or medical conditions. When hair cells are damaged, they may send aberrant signals to the brain. Sometimes tinnitus originates in the brain itself, with neural circuits becoming hyperactive in the absence of normal input, similar to phantom limb sensations. Scientists are still working to fully understand it, but tinnitus is a reminder that hearing is as much about the brain as the ear. Loud sounds can damage your hearing permanently. When hair cells in the cochlea are damaged or destroyed, they don't regenerate in humans. Unlike many animals, including birds and fish, which can regrow damaged hair cells throughout life, human hair cells are formed during fetal development and must last your entire lifespan. Each one you lose represents a tiny portion of your hearing that's gone forever. Prolonged exposure to sounds above 85 decibels, about the level of city traffic or a busy restaurant, can cause gradual damage. At 90 decibels, damage can occur after just 2 hours of exposure. At 100 decibels, roughly the level of a chainsaw or a loud concert, damage can occur in 15 minutes. At 120 decibels and above, even momentary exposure can cause immediate permanent damage. Sudden exposure to extremely loud sounds, like explosions, gunfire without hearing protection, or very loud music at close range, can cause immediate damage through direct mechanical trauma to the cochlear structures. The hair cells can be torn from their moorings, the basilar membrane can be damaged, and in extreme cases, the tympanic membrane can rupture. That's why hearing protection matters. Earplugs or earmuffs don't make you miss out on sound. They reduce the intensity to safer levels while still letting you hear what's important. And they preserve the hair cells you'll need for the rest of your life. Noise-induced hearing loss is one of the most common, yet most preventable forms of hearing impairment. Some people can hear their own eyeballs moving. It sounds strange, but it's a real condition called superior canal dehiscence syndrome. In people with this condition, there's a small opening or thinning in the bone covering one of the semicircular canals. This abnormal opening allows sounds from inside the body to be transmitted to the inner ear with unusual efficiency. People with superior canal dehiscence might hear their eyes moving in their sockets, their own heartbeat, their footsteps resonating in their head, or even the sound of their joints creaking. They may also experience vertigo or balance problems triggered by loud sounds or by straining. Humans can't use echolocation the way bats or dolphins do naturally, but we're not entirely without the ability. Blind individuals who've trained in echolocation can make clicking sounds with their tongues and use the returning echoes to navigate, detecting obstacles, identifying textures and shapes, and even estimating distances. With practice, they can navigate complex environments, ride bicycles, and perform tasks that would seem to require sight. Brain scans of expert echolocators show something fascinating. When they echolocate, their visual cortex lights up, even though they're not using their eyes. The brain is adapting, repurposing the neural real estate normally reserved for vision to process spatial information from sound instead. This demonstrates the brain's remarkable plasticity and its ability to use whatever information is available to build a model of the world. Your hearing begins before you're born. By around 18 to 25 weeks of gestation, the auditory system is functional enough that a fetus can hear sounds from outside the womb. The amniotic fluid and tissues muffle high frequencies, so what the fetus hears is dominated by low-frequency sounds like the mother's heartbeat, the gurgling of her digestive system, and the rhythm and intonation of her voice, though not the specific words. Loud external sounds like music or slamming doors also make it through. After birth, newborns show clear preferences for sounds they heard repeatedly before they were born. They prefer their mother's voice over other female voices. If a mother read a particular story or sang a particular song repeatedly during pregnancy, her baby will show recognition of that familiar sound. The world of sound welcomes you before you ever take your first breath, and those prenatal auditory experiences shape your early preferences and may even influence language development. Touch, our largest sensory organ. Touch is different from vision and hearing in a fundamental way. Those senses are concentrated in specific organs, your eyes and ears. Touch is spread across your entire body, woven into your largest organ, your skin. An average adult has roughly 20 square feet of skin, weighing about 8 pounds and making up approximately 16% of total body weight. Every square inch of it is packed with sensory receptors. Some parts of your body, like your fingertips and lips, have especially dense concentrations of touch receptors, with as many as 2,500 receptors per square centimeter. Other parts, like the middle of your back, have far fewer, sometimes as few as 50 receptors per square centimeter. This dramatic variation in receptor density is why you can detect fine details with your fingertips, but not with your elbow. Your skin contains several different types of touch receptors, mechanoreceptors, and each type responds to different stimuli. Meissner's corpuscles, located primarily in your fingertips, palms, lips, and other highly sensitive areas, respond to light touch and low-frequency vibration in the range of 10 to 50 Hz. They're especially good at detecting changes in texture and grip, and they let you feel textures and notice when something first makes contact with your skin. Meissner's corpuscles adapt quickly to constant stimulation, which is why you notice the initial touch, but then the sensation fades if the stimulus doesn't change. Merkel's discs, also concentrated in fingertips and lips, respond to sustained pressure and fine spatial details. They adapt slowly, which means they continue firing as long as pressure is applied. This sustained response is what allows you to maintain awareness of objects you're holding and to perceive fine details like the edges of a coin or the raised dots in Braille. Merkel's discs have the smallest receptive fields of any touch receptor, sometimes as small as 2 to 3 millimeters in diameter, which gives them exceptionally high spatial resolution. Pacinian corpuscles detect deep pressure and high frequency vibration in the range of 100 to 300 Hz. They're located deeper in the skin and also in muscles, joints, and internal organs. They're the most sensitive mechanoreceptors when it comes to vibration, able to detect oscillations with amplitudes as small as 10 nanometers. They're why you can feel vibrations through your bones and sense footsteps approaching on a wooden floor. Pacinian corpuscles adapt very quickly, responding primarily to changes in stimulation rather than sustained pressure. And Ruffini endings respond to skin stretch and sustained pressure. They're located deep in the skin and in joint capsules, where they contribute to proprioception by detecting the stretching of skin around joints. Like Merkel's discs, they adapt slowly, providing sustained signals about ongoing touch and skin deformation. These receptors don't work in isolation. Your brain combines their signals, integrating information from multiple receptor types and multiple locations to create the rich, multifaceted experience of touch. When you pick up a smooth stone, Meissner's corpuscles detect its coolness and texture during the initial contact. Merkel's discs feel its edges and shape continuously as you hold it. Pacinian corpuscles register its weight through deep pressure and any subtle vibrations as you shift it in your hand. Ruffini endings detect the stretching of your skin as your fingers curl around it. Together, they tell you everything you need to know about the stone without you ever looking at it. Temperature sensation, though often grouped with touch, is actually mediated by separate receptors. Your skin has thermoreceptors that respond specifically to warmth and cold. Cold receptors, which are actually responding to cooling rather than absolute cold, outnumber warm receptors by about 3 to 1 in most areas of skin. This is why you're generally more sensitive to decreases in temperature than increases, and why a room can feel uncomfortably cold even when it's only a few degrees below your preferred temperature. These thermoreceptors don't detect absolute temperature. Instead, they detect changes in temperature and rates of heat transfer. That's why a metal object feels colder than a wooden object at the same temperature. Metal conducts heat away from your skin much faster, creating a larger temperature gradient and a stronger signal from your cold receptors. It's also why a room feels cool when you first enter from outside on a hot day, but feels normal after you've been there for a few minutes. Your thermoreceptors adapt to the constant temperature and stop signaling strongly unless the temperature changes again. Pain is also separate from touch, though it travels through similar neural pathways. Your body has specialized pain receptors called nociceptors scattered throughout your skin, muscles, joints, and most internal organs. They respond to potentially harmful stimuli like extreme temperatures, intense pressure, tissue damage, or irritating chemicals. There are different types of nociceptors. Some respond to mechanical damage like cuts or crushing. Others respond to extreme heat or cold. Still others respond to chemical signals released by damaged or inflamed tissue. But pain is more complex than a simple alarm system. The signals from nociceptors travel to your spinal cord, where they can be modulated before even reaching your brain. Your spinal cord has circuits that can amplify or dampen pain signals based on other simultaneous inputs. This is the basis of the gate control theory of pain. When you rub a bumped elbow, you're activating touch receptors that can partially close the gate on pain signals, reducing the pain you feel. Once pain signals reach your brain, they're further interpreted and modulated by context, expectation, attention, emotion, and past experience. The same stimulus can feel more or less painful depending on your emotional state, what you expect to feel, what you're focused on, and what the pain means to you. This is why a paper cut you notice immediately seems to hurt more intensely than a scrape you discover only after seeing the blood. Your brain interprets and shapes the experience of pain in ways that go far beyond the simple presence or absence of tissue damage. Your sense of touch adapts remarkably quickly to constant stimulation. When you first put on a watch, a piece of jewelry, or clothes, you feel them clearly against your skin. After just a few minutes, you forget they're there. The receptors in your skin, especially the rapidly adapting ones like Meissner's and Pacinian corpuscles, stop responding to constant, unchanging pressure. This sensory adaptation is essential for normal function. Without it, you'd be constantly aware of every point where your body touches the surface, every thread of your clothing against your skin, every strand of hair touching your shoulders. The constant barrage of unchanging sensory information would be overwhelming and would make it difficult to detect new, potentially important stimuli. This adaptation is specific to particular locations and types of stimulation. If you adapt to the pressure of a watch on your wrist, you'll still feel a light touch on your arm. The system is constantly adjusting, filtering out the familiar and unchanging while remaining exquisitely sensitive to novelty and change. You can detect vibrations with incredibly subtle amplitudes. Research suggests that under ideal conditions, fingertips can detect vibratory displacement amplitudes as small as 2/10,000ths of an inch, or roughly 5 nanometers. That's far smaller than the wavelength of visible light and approaching the scale of individual molecules. This extraordinary sensitivity is why you can feel the vibration of your phone in your pocket, even through thick fabric, why you can sense the texture of even very smooth surfaces when you run your fingers across them, and why skilled craftspeople can detect minute imperfections in surfaces that look perfectly smooth. Your fingertips are covered in tiny ridges and whorls that create your unique fingerprints. These ridges, called epidermal ridges or friction ridges, serve multiple functions. They improve your grip on objects by increasing friction and channeling away moisture, but they also enhance your sensitivity to texture and vibration. When your finger moves across a textured surface, the ridges amplify the resulting vibrations, making it easier for your mechanoreceptors to detect fine details. The ridges essentially act as biological amplifiers, turning subtle surface features into more pronounced vibrations that your receptors can more easily detect. No two people have ever been found to have identical fingerprints, not even identical twins who share the same DNA. While genetics influences the general pattern of ridges, the specific arrangement is shaped by random factors during fetal development, including the precise position of the fetus in the womb, blood pressure, and the concentration of various chemicals in the amniotic fluid. The result is a pattern that's unique to you and remains stable throughout your life, even as your skin grows and ages. Some parts of your body are far more sensitive to touch than others, a phenomenon quantified by 2-point discrimination thresholds. If someone touches your fingertip with 2 points that are just 2 millimeters apart, you can tell that there are 2 separate touches rather than one. But on your back, those same 2 points would need to be 40 millimeters apart, or more, 20 times the distance before you could distinguish them. Your lips and tongue are almost as sensitive as your fingertips, with 2-point thresholds of 3 to 4 millimeters. Your palms are moderately sensitive at about 10 to 15 millimeters. Your shoulders and thighs require 30 to 40 millimeters. This variation in sensitivity is reflected in how your brain is organized. The somatosensory cortex, the part of your brain that processes touch information, devotes disproportionate amounts of space to processing signals from your most sensitive body parts. If you were to draw a human figure sized according to how much brain space is devoted to each body part, rather than actual physical size, you'd get a strange-looking creature called a sensory homunculus, with enormous hands, lips, and tongue, and a relatively tiny torso, arms, and legs. Phantom sensations are one of the strangest phenomena related to touch. After an amputation, many people, perhaps up to 80%, continue to feel their missing limb. They can feel it move, experience sensations of touch, pressure, or temperature on it, and often feel pain in it. This happens because the part of the brain that processes touch from that limb is still active. Without input from the limb itself, it sometimes generates signals on its own or responds to input from nearby body regions, creating vivid sensations that feel completely real. Interestingly, neurologist V.S. Ramachandran discovered that if you touch certain areas on an amputee's face or remaining limb, they might feel the touch in their phantom limb as well. This happens because in the sensory homunculus, the brain representation of the hand is adjacent to the representation of the face. When the hand area is deprived of normal input, neurons representing the face can expand their territory into the hand area, touch the face, and signals spill over into the former hand area, creating the sensation of being touched on a hand that's no longer there. This demonstrates the brain's remarkable plasticity and the extent to which your experience of touch is constructed by your brain rather than simply transmitted from your skin. Your skin is constantly sending signals to your brain about texture, pressure, temperature, pain, and more. Most of these signals never reach your conscious awareness. Your brain filters them aggressively, highlighting only the changes and the potentially important stimuli while suppressing the constant background sensation. The feel of your chair supporting you right now, the touch of your clothes against your skin, the temperature of the air on your exposed skin, all of these fade into the background, noticed only when they change significantly or when you deliberately direct your attention to them. Touch is deeply tied to emotion and social bonding through a special type of nerve fiber called C-tactile afferents, also known as CT afferents. Unlike the mechanoreceptors that send signals through fast myelinated nerve fibers, CT afferents are unmyelinated and carry signals much more slowly, at less than 2 meters per second. They're found primarily in hairy skin, like your arms and back, rather than in glabrous skin, like your palms. CT afferents respond optimally to slow, gentle stroking at speeds of about 3 to 5 centimeters per second, right around the speed of a gentle, affectionate caress. When activated, they don't send signals to the primary somatosensory cortex where other touch information is processed. Instead, they send signals to areas of the brain involved in emotion and reward, like the insular cortex and orbitofrontal cortex. This specialized system for pleasant social touch is thought to be important for bonding, comfort, and emotional well-being. A hug, a pat on the back, a gentle hand on the shoulder, holding hands. These touches activate CT afferents and communicate comfort, affection, and connection in ways that go beyond the purely physical sensation. The importance of touch for emotional and physical health, especially in infancy and early childhood, is well documented. Babies who receive insufficient touch can fail to thrive, even if their other needs are met. Your skin also remembers, in a sense. Repeated touch in the same location can lead to changes in receptor density and in how much of your brain's somatosensory cortex is devoted to that area. This is called cortical reorganization, and it happens throughout life. Musicians develop enhanced tactile sensitivity in their fingertips from years of practice. The motor cortex and somatosensory cortex regions representing their fingers expand, providing finer control and more detailed sensory information. People who read Braille develop extraordinary tactile discrimination in their reading finger, often the index finger of their dominant hand. The brain, in response to the increased importance of tactile information from that finger, devotes more neural resources to processing it. This plasticity works in both directions. If you stop using a particular skill, the brain regions devoted to it can shrink as the brain reallocates resources to currently relevant tasks. The brain is constantly optimizing its organization based on what you're actually using. Taste, chemical detection. Taste is one of your chemical senses, working by detecting specific molecules in the things you eat and drink. It's an ancient sense, one that helped your ancestors identify nutritious foods and avoid toxins. And despite what you might have learned in elementary school, there's no neat map of the tongue with different regions exclusively responsible for different tastes. That's a persistent myth based on a mistranslation of German research from the early 1900s. The reality is that all areas of your tongue that contain taste buds can detect all the basic tastes, though there may be subtle differences in sensitivity to different tastants in different regions. Those basic tastes number 5, not the four you might have learned. There's sweet, sour, salty, and bitter, the familiar quartet. But there's also umami, a savory taste often described as meaty, brothy, or deeply satisfying. It's the taste of glutamate, an amino acid that's particularly abundant in protein-rich foods. Parmesan cheese, ripe tomatoes, soy sauce, mushrooms, meats, and the Japanese seasoning MSG, monosodium glutamate, all have strong umami flavors. The Japanese scientist Kikanai Ikeda identified and named this taste in 1908, extracting pure glutamate from kombu seaweed. For decades, Western science was skeptical of umami as a distinct taste, but it's now recognized worldwide as the fifth basic taste, with dedicated receptors and neural pathways distinct from the other four. Your tongue is covered with small bumps called papillae, visible structures that give your tongue its characteristic texture. There are several types of papillae, each with different functions. The largest are the circumvallate papillae, typically 8 to 12 in number, arranged in an inverted V shape at the very back of your tongue. Each one is a couple of millimeters across, large enough to be easily visible, and each is surrounded by a deep groove that contains hundreds of taste buds. These papillae account for a significant proportion of your total taste bud count, despite their small number. Smaller fungi-form papillae, so named because they're shaped like tiny mushrooms, are scattered across the front 2/3 of your tongue, particularly dense on the tip and edges. Each one contains anywhere from 3 to 5 taste buds, on average. The variability in fungi-form papillae density accounts for much of the variation in taste sensitivity between individuals. Some people have dozens per square centimeter, while others have far fewer. And the most numerous, but smallest, are filiform papillae, which cover most of the tongue's surface and give it its roughness. Unlike other papillae, filiform papillae don't contain taste buds at all. Instead, they're mechanical structures that help manipulate food and provide tactile feedback. They're also the papillae that become more prominent when your tongue is coated, giving it a white or yellowish appearance when you're dehydrated or ill. Embedded within the circumvallate and fungi-form papillae, as well as in papillae on the soft palate, throat, and upper esophagus, are your taste buds. These are small, onion-shaped clusters of 50 to 150 receptor cells, along with supporting cells and basal cells that can develop into new receptor cells. An average person has between 2,000 and 8,000 taste buds, though the number varies considerably between individuals. Some people, called supertasters, have significantly more, while non-tasters have fewer. Each taste bud contains receptor cells for all 5 basic tastes, not specialized for just one taste. The receptor cells have hair-like projections called microvilli, extending from their tips into the taste pore, a small opening at the surface of the tongue. When you eat, molecules from your food dissolve in your saliva and reach these microvilli, where they interact with receptors on the cell surface. Different tastes are detected by different mechanisms. Sweet, umami, and bitter tastes are detected by G-protein-coupled receptors, proteins embedded in the cell membrane that trigger complex intracellular signaling cascades when they bind to their specific taste and molecules. Humans have roughly 25 different bitter receptors, far more than for any other taste, because there are many different types of toxic bitter compounds that need to be detected. Sweet and umami tastes each have dedicated receptor combinations formed by pairs of related proteins. Salty and sour tastes work differently. Salty taste is primarily detected by ion channels that allow sodium ions to enter the receptor cell directly, depolarizing it. Sour taste is detected by hydrogen ions, which can affect the cell in multiple ways, including blocking potassium channels. The mechanisms for salt and sour detection are still being researched, and there may be multiple pathways involved for each. When a taste receptor is activated, whether by direct ion entry or by a G protein coupled cascade, the receptor cell depolarizes and releases neurotransmitters onto the sensory nerve fibers that innervate the taste bud. These nerve fibers carry the taste information to the brain, traveling through 3 different cranial nerves, depending on the location of the taste bud. The taste information travels first to the brainstem, then to the thalamus, and finally to the gustatory cortex in the brain's insular and frontal operculum regions, where taste is consciously perceived. Your taste buds have a remarkably short lifespan. Individual taste receptor cells are replaced roughly every 10 to 14 days. This rapid turnover is necessary because these cells are directly exposed to the harsh chemical and thermal environment of your mouth. Food acids, hot beverages, abrasive textures, and the general wear and tear of eating all take their toll. The constant replacement ensures that you always have functioning taste receptors, though the turnover rate can be affected by various factors including age, nutrition, and exposure to irritants like smoking or extremely hot foods. But as you age, this replacement process slows down, and the total number of taste buds typically decreases. You're born with roughly 10,000 taste buds. By middle age, you might have 8,000. By older adulthood, you might have only 5,000 or fewer, concentrated more in the back of the tongue and soft palate, with fewer remaining on the tongue tip and sides. This is one of several reasons why older people often find that food doesn't taste as flavorful as it once did. The other major contributor is the decline in sense of smell, which provides most of what we perceive as flavor. The 5 basic tastes each serve an evolutionary purpose related to nutrition and safety. Sweetness indicates energy-rich carbohydrates, important for survival when calories were scarce. Saltiness helps you detect and maintain proper electrolyte balance, critical for nerve and muscle function. Umami signals protein, essential for building and repairing tissues. Sourness can warn you of unripe fruit, which might be difficult to digest, or fermentation, which could indicate spoilage, though fermented foods can also be safe and nutritious. And bitterness is often a sign of toxins, as many poisonous plants produce bitter alkaloids. Your strong aversion to bitter tastes is a protective mechanism, though it varies significantly between individuals. Interestingly, you're born with innate preferences and aversions for certain tastes. Newborn babies who've never tasted food before will display facial expressions of pleasure when given something sweet and expressions of disgust when given something bitter. These preferences are hardwired into your biology, shaped by millions of years of evolution. Sweet meant energy, bitter often meant poison. These survival critical associations are built in from birth, but taste preferences aren't entirely fixed. They can change significantly with experience and exposure. Many bitter foods like coffee, dark chocolate, bitter greens like kale and arugula, and certain vegetables like Brussels sprouts are acquired tastes. Repeated exposure can reduce your sensitivity to bitterness in those specific foods and even teach you to enjoy them. Your brain learns through experience that these bitter substances aren't actually harmful and can even begin to associate their bitterness with positive effects, like the alertness from caffeine or the satisfaction of a nutritious meal. Children typically find bitter foods more aversive than adults, and their preferences often shift toward accepting a wider range of flavors as they mature. Some people are genetically predisposed to be super tasters. They have significantly more fungiform papillae than average, sometimes twice as many, and consequently more taste buds. This makes them more sensitive to all tastes, but especially to bitterness. For super tasters, foods like black coffee, unsweetened chocolate, grapefruit, tonic water, and dark leafy greens can be almost intolerably bitter. They're also often more sensitive to the burn of capsaicin in chili peppers and to the fattiness of rich foods. Being a super taster might sound like it would make food more enjoyable, but it's often the opposite. The intensity of certain flavors can be overwhelming rather than pleasurable, leading super tasters to avoid many foods that others enjoy. On the other end of the spectrum are non-tasters, people with fewer taste buds, who are significantly less sensitive to tastes, especially bitterness. And in between are medium tasters, comprising the majority of people, with average taste bud, density, and sensitivity. The variation between these groups is partially genetic, influenced by variations in taste receptor genes, particularly the TAS2R38 gene, which affects sensitivity to certain bitter compounds. Your ability to taste is heavily influenced by your sense of smell, so much so that what you think of as taste is actually mostly smell. When you eat, aromatic volatile molecules from your food travel from your mouth up through the nasopharynx, the passage connecting the back of your mouth to your nasal cavity to reach your olfactory receptors. This pathway is called retronasal olfaction, and it's distinct from the normal orthonasal olfaction that occurs when you sniff. Retronasal olfaction is responsible for most of what you perceive as the flavor of food. The 5 basic tastes detected by your tongue provide only a simple framework. Smell fills in the enormous complexity and variety of flavors. This is why food tastes bland when you have a cold and your nasal passages are congested. Your taste buds are working perfectly fine, detecting sweet, salty, sour, bitter, and umami just as they normally would. But your sense of smell is blocked, and without it, you can't distinguish the rich flavor of strawberry from cherry, or chicken from beef, or cheddar cheese from Swiss. The richness, the complexity, the specific character that makes one food different from another comes almost entirely from smell. Temperature profoundly affects taste perception. Cold temperatures dull your sense of taste across all 5 basic tastes, though the effect is strongest for sweetness. This is why ice cream, straight from the freezer, tastes less sweet than slightly melted ice cream, even though the sugar content is identical. It's also why drinks often seem less sweet when they're very cold, and why room temperature soda can taste cloyingly sweet if you're accustomed to drinking it cold. The sugar content hasn't changed, but your perception of sweetness is temperature dependent. The optimal temperature for taste perception is generally considered to be between 20 and 35 degrees Celsius, roughly room temperature to body temperature. Spiciness, that burning sensation you get from chili peppers, isn't actually a taste at all. It's a pain signal, triggered by a molecule called capsaicin that binds to TRPV1 receptors, pain receptors that normally respond to heat. Capsaicin activates these receptors directly, tricking your nervous system into thinking your mouth is actually burning, even though there's no heat source. This is why spicy foods literally feel hot, and why the burn persists even after you've swallowed. Water doesn't help because capsaicin isn't water soluble. Milk, which contains casein protein that can bind to and wash away capsaicin, provides more relief. The burning sensation engages pain pathways and can trigger protective responses like increased salivation and sweating. Similarly, the cooling sensation of mint isn't a taste. Menthol, the active compound in peppermint and spearmint, triggers TRPM8 receptors, cold-sensitive receptors that normally respond to cool temperatures. Menthol activates these receptors directly, creating the sensation of coolness without any actual temperature change. Your mouth isn't getting colder. You're just receiving the same signal you'd get from something cold. touching your tongue. Your saliva is absolutely essential for taste. Taste molecules must be dissolved in liquid before they can reach your taste receptors. If your mouth is completely dry, you can't taste anything at all, regardless of how flavorful the food is. This is why people with conditions that reduce saliva production, like Sjögren's syndrome or side effects from certain medications, often complain of loss of taste. The problem isn't their taste buds themselves, but the lack of the aqueous medium needed to carry taste molecules to those receptors. Saliva also contains enzymes that begin breaking down food, and this breakdown can release additional taste and molecules, enhancing flavor. Some taste receptors can even detect the breakdown products specifically, contributing to the overall flavor experience. The composition of saliva varies between individuals and can be influenced by hydration, diet, and health status, all of which can subtly affect how food tastes. Interestingly, your sense of taste can be influenced by what you see. Studies have demonstrated that people perceive drinks to taste different when the color is changed, even when the actual flavor is identical. A red drink is expected to taste like cherry or strawberry, and people often report tasting those flavors even when they're completely absent. Similarly, white wine dyed red is often described using terms typically associated with red wine, even by wine experts. Your brain doesn't just passively receive taste information, it actively interprets it based on all available sensory cues, creating expectations that shape your experience. Context, expectation, and even price can influence taste perception. Wine tastes better when participants believe it's expensive, even when it's actually a cheap wine in a fancy bottle. Food tastes better when it's beautifully presented on elegant dishware. The same meal can be perceived as more delicious when eaten in a nice restaurant compared to out of plastic containers. Your brain integrates taste information with visual cues, social context, expectations, memories, and cognitive judgments, creating a holistic experience of flavor that goes far beyond the simple chemical detection happening on your tongue. Smell, the ancient sense. If taste is one of your chemical senses, smell is the other, and it's arguably the more powerful of the two. Your sense of smell, or olfaction, can detect an enormous range of molecules floating in the air around you. The exact number of odors humans can distinguish remains uncertain and is still debated by scientists. Earlier estimates suggested around 10,000 distinct odors, while a controversial 2014 study claimed over 1 trillion, though that claim has since been disputed on mathematical grounds. What's certain is that your olfactory system is extraordinarily sensitive and versatile, capable of detecting and distinguishing a vast array of scents. Smell begins in a small patch of tissue high up in your nasal cavity, behind the bridge of your nose. This is the olfactory epithelium, and it's covered with millions of olfactory receptor cells. Each cell has tiny hair-like projections called cilia that extend into the mucus lining your nasal cavity. When odor molecules dissolve in this mucus, they bind to receptors on the cilia. Humans have roughly 400 different types of olfactory receptors. Each type responds to a different range of molecular shapes and chemical properties. A single odor, like the smell of coffee or roses, is made up of dozens or even hundreds of different molecules. Your brain identifies the smell by analyzing the pattern of which receptors are activated and how strongly. The signals from your olfactory receptors travel directly to the olfactory bulb, a structure at the base of your brain. From there, they're sent to several areas, including the amygdala and hippocampus, brain regions involved in emotion and memory. This direct connection is why smells are so powerfully linked to emotions and memories. Unlike other senses, which are processed first by the thalamus Before reaching emotional centers, smell has a fast track to the parts of your brain that handle feeling and remembering. This is why a particular scent can instantly transport you to a specific moment in your past. The smell of a certain perfume might bring back memories of a grandmother. The scent of chlorine might recall childhood summers at a pool. These scent memories, sometimes called Proustian memories after the author Marcel Proust who wrote about them, can be vivid and emotionally intense. Your sense of smell is more sensitive in some ways than any laboratory instrument. You can detect certain odor molecules at concentrations as low as a few parts per trillion. Methyl mercaptan, the chemical added to natural gas to make it detectable, can be smelled at concentrations of just a few parts per billion. Your nose is essentially a highly sophisticated chemical analyzer, constantly sampling the air. Women generally have a more acute sense of smell than men. The difference isn't huge, but it's measurable. Women tend to score better on tests of odor identification and sensitivity, especially during certain phases of the menstrual cycle, when estrogen levels are high. Pregnancy can also heighten the sense of smell, sometimes dramatically. Many pregnant women report being able to detect odors they wouldn't normally notice, and finding certain smells intensely unpleasant. Your sense of smell adapts quickly. When you first enter a room with a strong odor, you notice it immediately. After a few minutes, you barely smell it at all. This is called olfactory adaptation. Your olfactory receptors become less responsive to a constant stimulus, effectively tuning it out. This allows you to detect new smells without being overwhelmed by background odors that aren't changing. But this adaptation is specific to particular odors. If you adapt to the smell of coffee, you'll still be able to detect the smell of flowers or smoke. Your olfactory system is constantly adjusting, filtering out the familiar and highlighting the novel. You lose olfactory receptor cells as you age, and unlike taste buds, they're not always replaced. By the time you reach your 70s, your sense of smell may be significantly diminished. This loss, called presbyosmia, can affect quality of life. Food becomes less appealing. It's harder to detect spoiled food or dangerous smells like gas leaks or smoke. The social and safety implications are real. Some people lose their sense of smell entirely, a condition called anosmia. It can be caused by head injuries, viral infections, nasal polyps, or neurodegenerative diseases. People with anosmia often report feeling disconnected from the world. They can't smell flowers, freshly baked bread, or the scent of loved ones. They can't detect smoke or gas leaks. And food loses most of its flavor, since smell is so integral to taste. Interestingly, partial loss of smell can be an early warning sign of neurodegenerative diseases like Parkinson's and Alzheimer's. The olfactory system is often affected before other symptoms appear. Researchers are exploring whether smell tests could be used as an early diagnostic tool for these conditions. Humans have a vomeronasal organ, a small structure in the nasal cavity that in many animals detects pheromones, chemical signals that influence behavior and physiology. In humans, this organ is present during fetal development, but is generally considered vestigial in adults. Whether humans can actually detect and respond to pheromones is still debated. Some studies suggest subtle effects, like changes in mood or hormone levels in response to certain chemical signals, but the evidence is far from conclusive. Your sense of smell is more active when you're hungry. Studies show that people rate food smells as more pleasant when they haven't eaten recently. This makes sense from an evolutionary perspective. When you need food, your sensory systems become more attuned to finding it. The same smell that's mildly pleasant when you're full can become intensely appealing when you're hungry. Different cultures have different odor preferences, shaped by experience and tradition. A smell that's pleasant in one culture might be neutral or even unpleasant in another. These preferences are largely learned, not innate. You develop associations between smells and experiences, and those associations shape how you perceive and react to odors throughout your life. Aromatherapy, the use of scents to influence mood and well-being, has been practiced for thousands of years. While some claims are exaggerated, there's evidence that certain scents can have real psychological effects. Lavender has been shown to have calming properties in some studies. Peppermint may increase alertness. These effects are likely mediated through the direct connections between the olfactory system and brain regions involved in emotion and arousal. Your ability to smell properly depends on clean air flowing through your nasal passages. Congestion from a cold, allergies, or a deviated septum can block odor molecules from reaching your olfactory receptors. This is why you can't smell well when you're sick. The receptors are fine, but the pathway to them is blocked. Interestingly, you can smell while you sleep, but your brain usually doesn't process those smells consciously. However, strong or significant odors can sometimes wake you. Smoke detectors are essential because while you might smell smoke in your sleep, you can't rely on smell alone to wake you from deep sleep. The hidden senses beyond the five. We talk about five senses: vision, hearing, touch, taste, and smell. But your body has several other sensory systems that are just as important, even though they don't get the same attention. Proprioception is your sense of body position and movement. Right now, without looking, you know exactly where your arms and legs are. You can touch your nose with your eyes closed. You can walk without constantly watching your feet. This is proprioception at work. Proprioceptors are sensory receptors located in your muscles, tendons, and joints. They detect the stretch and tension of muscles, the angle of joints, and the position of your limbs. Your brain integrates these signals to create a detailed map of your body's position in space. Without proprioception, even simple movements become incredibly difficult. There are rare cases of people who've lost proprioception due to nerve damage. They can move their muscles, but without constant visual feedback, they have no idea where their limbs are. Walking, picking up objects, even sitting still requires intense concentration and visual monitoring. Proprioception works unconsciously most of the time. Your brain is constantly tracking your body position and adjusting your movements automatically. When you walk, you don't consciously think about which muscles to contract or how much to bend your knee. Proprioception handles it in the background. Athletes rely heavily on proprioception. A gymnast knows exactly where every part of their body is during a complex flip. A basketball player knows the position of their hand and the ball without looking. Proprioceptive feedback is what allows for smooth, coordinated movement. Your vestibular sense is your sense of balance and spatial orientation. It's centered in your inner ear, in those semicircular canals we talked about earlier. These canals detect rotation of your head in 3 dimensions, and nearby structures called the utricle and saccule detect linear acceleration and the pull of gravity. Together, these structures tell your brain how your head is moving and oriented in space. This information is combined with visual input and proprioception to maintain your balance and sense of which way is up. When your vestibular system malfunctions, the results can be dramatic. Vertigo, the sensation that you or your surroundings are spinning, is often caused by problems in the inner ear. Even small disruptions can throw off your balance and leave you feeling disoriented and nauseated. Motion sickness occurs when there's a mismatch between what your vestibular system is sensing and what your eyes are seeing. When you read in a car, your eyes see a stationary page, but your vestibular system detects the motion of the vehicle. This conflict confuses your brain and can trigger nausea. The exact mechanism isn't fully understood, but the mismatch between sensory inputs is key. Interoception is your sense of your internal body state. It includes your awareness of hunger, thirst, need to use the bathroom, heart rate, breathing, and various other internal signals. You're usually not consciously aware of most interoceptive signals, but they influence your behavior and emotional state constantly. Some people have very strong interoception and are highly aware of their internal states. Others have weaker interoception and might not notice hunger or thirst until it becomes urgent. Research suggests that interoception is linked to emotional awareness. People who are more attuned to their internal bodily states tend to be better at recognizing and understanding their emotions. Thermoception is your sense of temperature. It's separate from touch, though we often group them together. Your skin has thermoreceptors that respond to warmth and cold, as we mentioned earlier. But you also have temperature sensors in your body's core that monitor your internal temperature and trigger responses like shivering or sweating to maintain a stable core temperature around 98.6 degrees Fahrenheit. Nociception is your sense of pain. Pain receptors, or nociceptors, respond to potentially harmful stimuli. There are different types. Some respond to intense mechanical pressure. Others respond to extreme temperatures. Still others respond to chemicals released by damaged tissue. Pain signals travel from nociceptors to your spinal cord and then to your brain, where they're interpreted as pain. But pain isn't just a straightforward signal. It's modulated at every level. Your spinal cord can amplify or dampen pain signals. Your brain can release endorphins that reduce pain. Your emotional state, your expectations, and your attention all influence how much pain you feel. This is why the same injury can hurt more or less depending on the circumstances. A soldier injured in combat might not feel pain until after the fight is over. Someone anxious about pain might feel it more intensely. Pain is a subjective experience, shaped as much by psychology as by physiology. There's also a sense called chronesthesia, sometimes called mental time travel. It's your ability to be aware of your existence in subjective time, to remember the past and imagine the future. While this might seem more cognitive than sensory, some researchers argue it's a distinct sense that allows you to perceive yourself moving through time. Your body also has receptors for detecting chemical changes in your blood, like oxygen and carbon dioxide levels. These chemoreceptors trigger automatic adjustments to your breathing and heart rate, keeping your blood chemistry balanced without any conscious effort from you. All of these hidden sensors work together constantly, providing your brain with a continuous stream of information about your body and your environment. You're not aware of most of it, but it's there, shaping your experience and guiding your actions every moment. Sensory integration and synesthesia. Your senses don't work in isolation. Your brain is constantly integrating information from multiple senses, weaving them together into a unified experience of reality. When you watch someone speak, you're not just hearing their words, you're also seeing their lips move, and your brain combines these audio and visual cues. This integration is so seamless that you don't notice it, but it can be demonstrated with clever experiments. The McGurk effect is a famous example. If you hear the sound bar while watching a video of someone's lips making the shape for ga, you'll often perceive a completely different sound, like da. Your brain is trying to reconcile the conflicting audio and visual information, and it creates a perception that's different from either input alone. This shows that what you hear isn't just determined by your ears. It's determined by the combination of all available sensory information. Your brain is constantly cross-checking different senses, looking for patterns and coherence. Sensory integration starts in infancy. Babies learn to coordinate what they see with what they touch and hear. When an infant reaches for a rattle, they're learning to connect the visual image of the rattle with the tactile sensation of grasping it and the sound it makes when shaken. Over time, these separate sensory streams become bound together into a unified perception of rattle. For most people, sensory integration happens automatically and unconsciously. But for some individuals, this integration doesn't work smoothly. Sensory processing disorder is a condition where the brain has trouble organizing and responding to sensory information. Someone with sensory processing disorder might be overwhelmed by textures, sounds, or lights that others find normal. Or they might seek out intense sensory experiences, craving strong stimulation. Synesthesia is a different phenomenon where stimulation of one sense triggers an experience in another sense. For someone with synesthesia, numbers might have colors, sounds might have tastes, music might evoke specific visual patterns. The most common form is grapheme color synesthesia, where letters and numbers are consistently associated with specific colors. Someone with this type of synesthesia might always see the number 5 as red or the letter A as blue. These associations are consistent over time and often date back to childhood. Sound color synesthesia, or chromesthesia, is another common type. Musical notes, chords, or entire pieces of music trigger specific color perceptions. The composer Olivier Messiaen had chromesthesia and incorporated these color associations into his compositions. Less common but equally fascinating is lexical gustatory synesthesia, where words evoke taste sensations. Someone with this condition might taste chocolate when they hear the word chair, or taste mint when they hear a particular name. Synesthesia is thought to involve increased cross-activation between different sensory areas of the brain. In most people, these areas are somewhat separated, with clear boundaries. In synesthetes, there may be more connections or less inhibition between areas, allowing activity in one area to spill over into another. Synesthesia runs in families and appears to have a genetic component. It's more common than was once thought, affecting perhaps 4% of the population to some degree. Many synesthetes don't realize their experience is unusual until they learn that others don't share it. For most synesthetes, their cross-sensory experiences are pleasant and enriching. Colors for numbers might make math more intuitive. Musical colors might make music more vivid. But in some cases, synesthesia can be overwhelming or distracting. Synesthesia is distinct from metaphor. When you describe music as bright or a voice as warm, you're using metaphorical language. But synesthetes aren't using metaphors. They're describing genuine perceptual experiences, triggered by cross-sensory connections in their brains. Interestingly, psychedelic drugs can sometimes induce temporary synesthesia-like experiences in people who don't normally have them. This suggests that the neural pathways for synesthesia exist in everyone, but they're normally suppressed or inactive. Beyond synesthesia, there are many ways that senses influence each other. The sound of food crunching affects how fresh you perceive it to be. The color of a room can influence how warm or cool you feel. The weight of a container affects how you judge the value of its contents. Your senses are in constant dialogue, each informing and modulating the others. Sensory extremes and records. Human senses have limits, but those limits are often more impressive than you might think. And when you compare human senses to those of other animals, you get a sense of both our capabilities and our constraints. Your vision covers an enormous range of brightness. The dimmest light you can see in complete darkness is just a few photons hitting your retina. The brightest light you can tolerate is roughly 1 billion times more intense. This range, from starlight to sunlight, spans about 10 orders of magnitude. Few artificial senses can match this dynamic range. Your hearing is similarly impressive. The quietest sound you can hear is roughly 0 decibels, a sound pressure of about 20 micropascals. The loudest sound you can tolerate before pain and damage occur is around 120 to 130 decibels. That's a ratio of 1,000,000 to 1 in sound pressure. Your sense of smell can detect certain molecules at incredibly low concentrations. Methylmercaptan, as mentioned, can be detected at just a few parts per billion. Some research suggests that for certain odor molecules, you might be able to detect concentrations as low as 1 part per trillion. That's 1 molecule mixed with 1,000,000,000,000 molecules of air. Your skin can detect vibrations with amplitudes smaller than a single atom's diameter. Your tongue can distinguish temperature differences of less than 1 degree Celsius. Your proprioceptive system can detect joint angle changes of less than half a degree. But compared to many animals, human senses are fairly modest. Eagles have vision roughly 4 to 8 times sharper than humans. They can spot a rabbit from over 2 miles away. Mantis shrimp have 16 types of color receptors compared to our 3, allowing them to see colors we can't even imagine. Some insects can see ultraviolet light. Some snakes can see infrared. Dogs have a sense of smell that's 10,000 to 100,000 times more sensitive than humans. They have roughly 300 million olfactory receptors compared to our 6 million. Bloodhounds can track a scent trail that's days old. Dogs can be trained to detect cancer, low blood sugar, and oncoming seizures through scent alone. Bats and dolphins use echolocation with such precision that they can detect objects smaller than a millimeter in complete darkness. Elephants can detect seismic vibrations through their feet, communicating with other elephants miles away through vibrations in the ground. Sharks can detect electrical fields, sensing the tiny electrical signals produced by muscle contractions in their prey. But humans have our own sensory strengths. Our color vision is excellent compared to most mammals. Our binocular vision and depth perception are superb, a legacy of our tree-dwelling ancestors. Our hands have extraordinary tactile sensitivity, allowing for fine manipulation and tool use. And our sensory systems are exceptionally well integrated with our cognitive abilities, allowing us to analyze, interpret, and make complex decisions based on sensory information. Sensory deprivation experiments have shown how much your brain relies on constant sensory input. When people are placed in environments with minimal sensory stimulation, darkness, silence, no touch, they often begin to hallucinate within hours. The brain, accustomed to processing a constant stream of sensory data, begins to generate its own inputs when external stimulation is removed. Conversely, sensory enhancement is possible. Through training and practice, sommeliers develop extraordinary abilities to detect subtle differences in wines. Perfumers can distinguish thousands of distinct scents and identify individual components in complex blends. Musicians develop acute pitch discrimination. Radiologists learn to see patterns in X-rays that untrained observers would miss entirely. These enhancements aren't just about the sensory organs themselves. They're about how the brain processes sensory information. With training and attention, your brain can learn to extract more information from the same sensory inputs, refining your perception far beyond your baseline abilities. Development and aging of senses. Your sensory systems don't spring into existence fully formed. They develop gradually, starting before birth and continuing to mature throughout childhood and adolescence. Your sense of touch develops first, in the womb. By 8 weeks of gestation, a fetus responds to touch around the mouth. By 14 weeks, the entire body surface is responsive to touch. This is one of the primary ways a fetus learns about their body and the environment of the womb, hearing develops next. By around 18 to 25 weeks, a fetus can hear sounds from outside the womb. The mother's heartbeat, her voice, and external sounds like music all reach the developing ear. Newborns show clear preference for their mother's voice and for sounds they heard repeatedly before birth. The auditory system is functional at birth, though it continues to refine over the first year of life. Taste and smell are functional before birth as well. The amniotic fluid carries flavors from the mother's diet, and the fetus can taste and smell yardis. Studies have shown that babies show preference after birth for flavors they were exposed to in the womb. If a mother eats a lot of garlic during pregnancy, her baby is more likely to accept garlic-flavored foods after birth. Vision develops last and is the least mature sense at birth. Newborns can see, but their vision is blurry, roughly 20/400. They can't focus on objects more than 8 to 12 inches away, about the distance from a nursing baby to the mother's face. They can detect movement and contrast, but can't see colors clearly yet. Over the first few months, vision improves rapidly. Color vision develops. The ability to track moving objects improves. Depth perception begins to emerge. By 6 months, most of the basic visual abilities are in place, though fine-tuning continues for years. Your senses reach their peak performance at different times. Hearing is sharpest in your 20s. Vision generally peaks in your late teens to early 20s. Taste and smell remain stable through early adulthood. Touch sensitivity varies by body part, but generally remains stable until middle age. But from your 20s onward, most sensory abilities gradually decline. High-frequency hearing is usually the first to go. By your 30s, you've likely lost the ability to hear sounds above 16,000 hertz or so. This is why teenagers can hear mosquito ringtones that adults can't. Vision changes too. The lenses in your eyes become less flexible, making it harder to focus on close objects. This is presbyopia, and it's why most people need reading glasses by their 40s or 50s. Color perception can shift subtly as the lens yellows with age, filtering out some blue light. Taste and smell decline significantly with age. You lose taste buds and olfactory receptor cells. The ones that remain become less sensitive. This is why older adults often complain that food doesn't taste as good as it used to. It's not nostalgia or the food itself. Their sensory systems are genuinely less sensitive. Touch sensitivity decreases too, especially in the extremities. The density of touch receptors decreases, and the nerves that carry touch signals become less efficient. This can affect balance and fine motor control. But not all sensory decline is inevitable. Some of it is due to damage accumulated over a lifetime. Loud noise exposure damages hearing. UV light exposure damages vision. Smoking impairs smell and taste. Protecting your senses throughout life can preserve them better into old age, and the brain has remarkable plasticity. Even as individual sensory abilities decline, the brain can compensate to some degree. Older adults often rely more on context and expectations to fill in sensory gaps. They might not hear every word in a conversation, but they can infer meaning from context. They might not see fine details, but they can recognize faces and navigate familiar environments. Keeping your senses active and engaged appears to help maintain them. People who continue to challenge their sensory systems through music, reading, cooking, and staying socially engaged tend to maintain better sensory function than those who withdraw from sensory-rich activities. The future of sensory science. Our understanding of the senses is constantly evolving. New technologies are revealing aspects of sensory processing we never knew existed, and applied research is creating ways to restore, enhance, and even create new sensory experiences. Cochlear implants have already restored hearing to hundreds of thousands of people with severe hearing loss. These devices bypass damaged hair cells in the cochlea and directly stimulate the auditory nerve with electrical signals. While they don't perfectly replicate natural hearing, they allow many profoundly deaf individuals to perceive sound and, with training, understand speech. Retinal implants are doing something similar for vision. These devices use cameras to capture images and translate them into electrical signals that stimulate remaining functional cells in the retina or the optic nerve. The resolution is still low, but even crude visual information can dramatically improve the quality of life for people who are blind. Sensory substitution devices are even more creative. These tools convert information from one sense into another. A system might use a camera to capture visual information and convert it into tactile patterns on the tongue or auditory signals. The brain learns to interpret these new patterns as visual information, essentially seeing through a different sense. Blind individuals using these devices have reported being able to navigate obstacles, recognize objects, and even read letters after training. The brain's plasticity allows it to repurpose areas normally used for one sense to process information from another. Virtual reality and augmented reality technologies are creating increasingly convincing sensory experiences. VR headsets combine high-resolution visuals, spatial audio, and haptic feedback to create immersive environments that can feel remarkably real. As these technologies improve, the line between virtual and real sensory experiences may become increasingly blurred. Brain-computer interfaces are perhaps the most ambitious frontier. These systems can read neural signals directly from the brain and use them to control devices or generate sensory feedback. Early versions are already allowing paralyzed individuals to move robotic limbs with their thoughts. Future versions might allow for direct sensory communication, sending information straight to the brain without going through the sensory organs at all. Research into the mechanisms of sensory processing continues to reveal surprising complexity. Scientists are discovering new types of sensory receptors, new pathways in the brain, and new ways that different sensory systems interact. Each discovery raises new questions and opens new possibilities. Gene therapy may someday be able to repair or replace damaged sensory receptors. There's already research into using gene therapy to restore hearing by regenerating hair cells in the cochlea, something that doesn't happen naturally in humans, but does in some other animals. Pharmaceuticals that enhance sensory processing or protect sensory organs from damage are in development. Drugs that could slow age-related hearing loss, improve night vision, or enhance olfactory function are all areas of active research. There's even speculation about creating entirely new senses. Could we develop devices that let humans detect magnetic fields like some birds do? Or sense electrical fields like sharks? Or perceive infrared radiation like snakes? These might sound like science fiction, but the underlying principles are sound. If you can convert a signal into a form the brain can process, the brain can learn to interpret it. The ethical implications of sensory enhancement are already being debated. If we can enhance hearing beyond normal human levels, should we? What about creating supervision or designer senses? These technologies could reduce inequality by helping those with sensory impairments. But they could also create new forms of inequality if only some people have access to enhancement. As we learn more about how the brain processes sensory information, we're also learning more about consciousness itself. Perception is not passive reception of external reality. It's an active construction, built by the brain from sensory inputs and shaped by expectations, memories, and attention. Understanding the senses is inseparable from understanding the mind. Closing reflection, and here we are, having wandered through the landscape of sensation, from the photons striking your retina to the vibrations rattling your eardrum, from the molecules dissolving on your tongue to the pressure waves rippling through your skin, from the chemical signatures floating through your nose to the hidden sensors that track your body's position and internal state. Every moment, your sensory systems are translating the physical world into electrical signals, and your brain is weaving those signals into the rich experience of being alive and aware. The redness of an apple, the softness of a blanket, the melody of a song, the scent of rain. None of these exist in the physical world. They exist in your mind, constructed from patterns of neural activity triggered by sensory input. You are not a passive observer of reality. You are an active participant in creating the reality you experience. Your senses are the interface between the world outside and the world inside your head. And that interface is far more complex, far more sophisticated, and far more fascinating than we often appreciate. You've learned tonight about the millions of receptor cells in your eyes, the thousands of taste buds on your tongue, the hundreds of types of olfactory receptors in your nose. You've heard about hidden senses like proprioception and interoception that most people never even think about. You've discovered that pain is as much psychological as physical, that smell is more tightly linked to memory than any other sense, and that what you think of as taste is mostly smell. You've explored synesthesia, where the boundaries between senses blur. You've considered how your senses develop before birth and decline with age. And you've glimpsed the future possibilities of sensory restoration, enhancement, and even creation. But perhaps most importantly, you've been reminded of something easy to forget in the rush of daily life. That the simple fact of perceiving the world is a profound and intricate miracle. The warmth of sunlight on your skin, the sound of laughter, the taste of your favorite food. These aren't just passive receptions. They're the result of billions of years of evolution, millions of specialized cells, and unimaginably complex neural processing happening in your brain right now. And all of this, every bit of it, is happening to you right now as you rest. Your senses are still working, quietly gathering information. Your skin is feeling the surface beneath you. Your ears are processing the ambient sounds around you. Your eyes, even closed, are detecting the quality of light. Your proprioceptors are tracking your body's position. Your interoceptors are monitoring your breath, your heartbeat, the subtle rhythms of your internal world. You are a sensing being, and that is a beautiful thing. If you've enjoyed this journey through the senses, if it's given you something gentle to think about as you drift towards sleep, please consider liking this video and subscribing to Whispered Knowledge. It helps other curious minds find their way here, and it lets me know you'd like more of these calm, thoughtful explorations. Thank you for spending this time with me tonight. Sleep well, rest deeply, and when you wake, perhaps you'll notice your senses just a little bit more. The colors might seem richer, the sounds more textured, the world more vivid. Because now you know, just a little more deeply, what an extraordinary thing it is to sense anything at all. Good night.